so much for that wonderful introduction. And as Professor DeBauer said, I work with the Center for Academic Research and Training in Anthropology, CARTA, some of you may be familiar with, and also with the Institute for Neural Computation at UCSD, which is an NIMH-funded training program that teaches doctoral students more about neurodevelopmental disorders and integrates that as a part of their research training. So I'd like to thank you all for joining me here today on this day that I'm sure for a lot of you is a day of really great uncertainty. And it means a lot to me to see a lot of you sitting here today. And perhaps today, more than any other day, my core message becomes even more important, which is to say that our evolutionary history has yielded a really remarkable species that is capable of great empathy and cognitive flexibility, and that our greatest strength has always been and will always be our diversity and our resilience. So as the last speaker in this wonderful series, I hope to make it so you can all sort of leave here today with a renewed sense of optimism about our lives and about our brains and how we fill this sort of broad spectrum of human cognitive potentials that enriches our lives as a particularly social and empathetic species. So I identify as both an anthropologist on the one hand and a neuroscientist on the other. And for the former, the central question in anthropological research has always been, what makes a human a human? So what is it that sets us apart from our closest living relatives, and how has our evolutionary history shaped us as a species? As you can see in the above cladogram, we share a branch of the evolutionary tree with a specialized group of primates. And some 6 million years of primate evolution separate us from our closest living relatives on this tree, the chimpanzees. And in this relatively short span of time, a lot has happened. You can compare the 6 million years to rats and mice, who are separated by some 12 million years of evolution, despite the fact that they're morphologically very similar, and many of you probably couldn't even tell them apart. So 6 million years separate us from our closest living relatives, and a lot of changes have taken place in our physiology and our behavior that set us apart as sort of a uniquely weird and wonderful species. So what is it that's so special about a human? And the first and most obvious of these fundamental traits to emerge that make us different was bipedalism, or this upright posture and walking that we all do. And millions of years ago, we emerged from the trees and began to lose some of our specialized physical and morphological traits that allowed for arboreal life, instead gaining a more forward-facing pelvis, long legs, and for some of us, a great deal of back pain. So we stand upright on 2 feet, shifting our weight from one leg to the other, sometimes over great distances, even sometimes running across the landscape in pursuit of prey. And this has also freed up our hands, and by freeing our hands, we've begun to use them differently in ways that have shaped this other fundamental set of features that makes us particularly unique. In addition to this upright walking, the other characteristic that seems to make us a very weird primate can be found in our other cognitive adaptations and specializations. So if you look around this room, our habitat has been extensively modified and artificially constructed in ways that no other species would ever dare to attempt. I'd encourage you to take a moment to consider the amazing excesses of our endeavors. Our computers and our cell phones, this building, this podium, these chairs, none of these are essential for survival, but are an extension of ourselves into the material world. Historically, culture has been defined by archaeologists as the extrasomatic means of adaptation. So that is, all of the adaptations that exist outside of our bodies and our physiology that are made by us to fulfill some kind of purpose. But the thing is, no one actually needs the latest MacBook or iPhone to survive. We as a species tend to go really overboard with the things that we create. The philosopher Arthur Koestler got it correct when he said in his 1967 book, The Ghost in the Machine, that the evolution of the brain not only overshot the needs of prehistoric man, it is the only example of evolution providing a species with an organ that it doesn't know how to use. And I like this quote for 2 reasons. First, it highlights the absolute absurdity of the excess to which our cognitive faculties have outpaced our survival-based needs. Second, it exposes us as utterly unprepared to make use of the gifts that evolution has given to us. And that is that we all need to learn how to make use of our talents and our skills. So what, if anything, makes human cognition so unique? Culture exists in the material world outside of us, and the technologies that we create are a logical extension of all of those objects and concepts that have preceded them before. So this is good news, because if we had to reinvent the stone tool every generation, there wouldn't be much hope for more sophisticated developments. The very first stone tools were quite simple by our standards, consisting of coming about some 3 million years ago as these sort of simple cores built together with a crude sharpened point that are created by bashing rocks together in a predetermined fashion. But it would take another 1.5 million or so years to raise the bar to the level of these more sophisticated hand axes, which we see here, that required a great deal more forethought to construct using bifacial retouching methods that honed it into a superior tool for a wide range of uses. Now, the glorious success of human cognitive ability is exemplified by the utilitarian but wholly remarkable advancement of things like the Acheulean hand axe in the Paleolithic that extend all the way into the advancement of hunting technologies such as the atlatl in a relatively long period of time. And this, of course, led to less danger in long-range pursuit of game and less potential for severe bodily injury, increasing survival and potentially helping us with hunting. But this, of course, was followed much later by a huge variety of totally unnecessary and really interesting inventions. The rapid ratcheting up of our technologies includes a lot of efforts that seem to seek to take human effort out of the equation, starting with Eli Whitney's invention of the cotton gin, all the way up to Henry Ford's assembly line and to the Model T, all the way into the sort of look mom, no hands appeal of Tesla's very first self-driving cars. Even in California, we don't need cars. We don't actually need these things to survive. But the best tools might win us some sort of social capital, increasing our potential for survival and, importantly, reproduction. So culture isn't entirely material, however, and as I insinuated, our social behavior additionally exhibits some sort of scaled up, massively exaggerated nature of human excess that it has defined us as a species. Cooperation and sociality are seen in varying degrees among primates, and we know that chimpanzee politics is fascinatingly complex, and that among chimpanzees, groups of adults will conduct raids and patrols on a small scale in a manner described in the literature by some as warfare. This, of course, pales in comparison to the massive and destructive efforts that humans are capable of. So just like our closest living relatives, however, we are also capable of great empathy and pro-sociality. And we can come together to build great nations and empires with sophisticated methods for governance on a large scale for the greater good, however difficult and fraught these processes might sometimes be. It's arguable, of course, that none of this would be at all possible had we not developed novel forms of communication, known as language. All social species have their own methods of communicating, but language is something that truly sets our species apart. We know from comparative anatomy that chimpanzees and other primates fundamentally lack the physical characteristics that make speech possible. So if we look at the diagram here, we can tell that there are some elements that would make the production of a range of sounds on the order of language very difficult. So the motor apparatus that allows for this articulation of this range of sounds and syllables, known as words, known as language, is uniquely derived in humans. And indeed, a really large amount of coordination between the mouth and the brain is required to make language happen. So since the two are so tightly linked, the degree to which the performative elements of language allows for the cognitive capacities associated with language should be considered. So in addition to our mouths, our brains seem to also be really important for all of the facets of human cognition that make us such an unusual and weird species. So what is it that actually makes the human brain unique? The most obvious difference between our brains and those of our closest living relatives is one of size. So compared to our closest living relatives, the chimpanzees, the human brain is 3 times larger in absolute size. We also have the largest brains relative to our body sizes of any other primate, with a brain around 1,000 to 2,000 cubic centimeters as compared to around 350 to 400 cubic centimeters in chimpanzees. And here you can see the line of what would be expected of human brain size relative to body size. Humans fall well outside the line of what you would expect. So we are part of a group known as the hominids that encompass a wide variety of taxa and a really interesting evolutionary history. So keep this in mind as we're talking about the various time points when all of these different adaptations have arisen. So the human brain has seen this threefold increase in size comparatively late in the game with regard to our evolutionary history, with Australopithecines having brains that are roughly the size of chimpanzees. And this gradual increase in size has given way to the rapid expansion significantly after the emergence of our genus Homo. So we've had big brains since before we were human, in fact. As you can see here, many fossil forms exhibit cranial capacity that falls well within the range of modern human variation. And Neanderthals, as it so happens, actually had the largest brains of all, on average, and yet that didn't save them from extinction, although some of their genetic material lives on in our own legacy. So between species, we can generally accept the overall increase in relative brain size corresponds roughly to increased intelligence because of the general energetic demands that brain tissue entails. If you have a bigger brain, you must be doing something with it. And this is something that we can just generally assume. So within a species, this correlation doesn't seem to hold up very consistently. So the correlation between brain size and intelligence within a species is not necessarily consistent. We know that on average, women tend to have smaller brains than men, but this tends to be because women overall tend to be smaller than men. And of course, this is a trend and not an absolute, and there's a broad range of variation, but we also know that intelligence between men and women isn't variable to the extent that it would constitute a difference in brain, or that it would correspond to a difference in brain size. So we also know that brain size is a poor predictor of individual intelligence, except in extreme cases of pathology where you have an unusually small brain that might coincide with developmental delays. And overall, brain size is not a predictor of intelligence within a species, but it's also not a predictor of intelligence within an individual. So as a case in point, physicist Albert Einstein, seen here, had a brain very much on the lower range of what would be expected, and a modest 1,230 grams, despite the fact that he was regarded as one of the great geniuses of our time. So we know that size matters between taxa, but how much does it matter? And can we attribute all of our remarkable capacities to just a gross increase in raw materials in terms of brain stuff? In other words, is the human brain simply a scaled up primate brain? On the left, you can see a dorsal view of the brains of a wide variety of mammalian species, and you may notice that some of them look bumpier than others. The folds that are called gyri in the cerebral cortex on the outside of the brain are actually caused by simple laws of physics. If you can imagine that the cortex is a sheet of cells called neurons that are all stacked together in layers, which it is, these folds allow you to pack more cortex into the skull. So some surprisingly gyrified animals include things like bears and giraffes, which exhibit this more gyrified appearance than the humble baboon brain that you see on the top row. So gyrification may or may not be a good indicator of species complexity and intelligence. Now, furthermore, if you take out the entire brain and you stick it in a specialized blender and you homogenize the whole thing and you count the individual neurons to predict the absolute number of neurons in a brain of any given species, scaling rules seem to predict that the human brain does not actually have more neurons than would be expected for a brain of its size. And it's important to note that this doesn't tell us much about how the relationships and connections between all of those neurons might vary importantly, and how they might be specialized to function in different ways in the brain. So for that information, we need to dig deeper and to conceptualize what role reorganization might have had in fueling cognitive complexity. So what about reorganization? When we're trying to understand what changes took place in the brain and how they underlie the peculiarities of human cognition, we look for characteristics that might be considered uniquely derived. In other words, that means that the elements of morphology and anatomy have seen differential modification as a function of our evolutionary history. And it's important to consider that these changes can occur at any number of different levels of investigation. So as shown in the diagram at right, these levels can be broken down from the largest to the smallest level of investigation, and changes may occur at each level that bear investigation for understanding their implications for informing our evolutionary history. And additionally, we must consider the quality of the information along two different lines of evidence, which can include direct evidence of human evolution and indirect evidence utilizing comparative methods. Let's talk about direct evidence. And when I say direct evidence, I'm referring to the process of biological evolution and the information that can only be derived from the fossil record. As seen here, a variety of changes to the size and the shape of fossil crania have been discovered that show the gradual increase in brain size and the modifications to the shape of the brain. So in letter A, you have a composite of modern chimpanzee crania, since the fossil record is actually very poor for non-hominid apes. So we have chimpanzees, and we are comparing these to the Australopithecines, where we see evidence for changes to the face and to its relationship with the cranium that suggests certain reorganizations in the brain case, and thus presumably the brain itself. Furthermore, we can observe differences in the shape between how our brains and the Neanderthal brains looked, and we can sort of appreciate how bulbous our heads look and how round they are and how interesting that is for a variety of reasons. So in terms of direct evidence, again, we can additionally examine the interior surface of the crania of all of these fossil hominid species and generate an endocast using either latex in a very crude method or some super advanced computerized CT scanning and 3D modeling techniques. And this up here is an example of a latex endocast, which is made from the inside of a skull of a modern human, the modern human depicted on the left. Now, as you can see, a lot is lost in terms of the brain and its translation, the surface of the brain to the skull. Since in life, the brain itself is actually surrounded by fluid and tissues that protect it from damage. You can also observe several few key surface features that are informative for understanding things like drainage patterns and for the general organization of the lobes of the brain. And of course, you can observe cranial capacity, which varies consistently throughout the fossil record and gradually increases. So in development across the lifespan, shape also seems to be of vital importance, as is the pattern of brain growth itself. And we know that compared to chimpanzees, human infants are born with a much lower percentage of total brain growth achieved. So some 50% of brain size is attained at birth in chimpanzees, as compared to around 36 to 40% of brain size attained at birth in humans. Humans additionally retain a slower rate of growth and maturation known as neoteny that allows for a greater percentage of this brain growth to occur during a time when social interactions can better shape the information and the formation of connections that occur between cells. So with this knowledge, we can also observe juvenile fossil specimens. So when we have fossil specimens of subadult individuals, we can use these to understand when in human evolutionary history these shifts in growth patterns have occurred and try to compare them to the material evidence for social and cognitive complexity observed among these different extinct species. So with regard to shape, we can also observe that fundamental shifts in the way the brain changes across development have occurred that seem to correspond to the reality that humans retain this larger, rounder, more juvenile shape into adulthood that relates to the expansion of the parietal lobes in the brain. For everything that extends beyond surface features of the brain, we have to utilize certain indirect methods, because as we know, brain tissue doesn't fossilize. It would be great if it did, but it doesn't. So seen here, we have the brains of several mammal species. And as you can tell, we don't have the largest brains in absolute terms. Cetacean brains, like the bottlenose dolphin depicted here, are large and complex, which relates sort of to the demands of the aquatic environment and long-range travel across different oceans and areas. And we can also observe the complexity of the cortical gyri, which are very, very complex in cetaceans, as well as in humans, chimpanzees. So as shown here in blue, the cerebral neocortex, this outer part of the brain, occupies a much larger percentage of the total volume of the human brain as compared to the monkey and rat depicted. And I'd also like to point out that there's this weird little organ at the front of the mouse and the monkey brains that seems to be absent in the humans. Now, what it actually is the olfactory bulb. And in humans, the olfactory bulb is super, super tiny, and it's tucked up against the frontal cortex. And as we know, our sense of smell is not our primary way of getting around in the world. Some of us smell bad, but all of us smell kind of poorly compared to other mammalian taxa. And this is because other sensory systems instead have taken primacy. Specifically, visual systems. In primates, our visual system has been expanded to fulfill the demands of visual predation and conspecific recognition and other heavily visual tasks that are associated with primate life. And this has led to certain modifications to the brain's internal circuitry, including an expanded lateral nucleus of the thalamus, which relates to relaying visual sensory information, as well as this expanded network of visual cortices in the cerebral cortex that help to encode and process this information. So how do we understand what the brain does? There are some fundamental lines of evidence that we use to understand what happens when something goes wrong. So we understand the function primarily through dysfunction. And this can include things like animal lesion studies, wherein you focally damage an area and try to predict what happens, and try to manipulate that, and try to figure out what will happen when you damage a part of the brain. The thing is, we can't really do this in humans. It's not considered ethical for whatever reason. So accidental lesion studies happen. And by that I mean that when natural injuries happen to the brain, these are really valuable for looking at neurodegenerative conditions and processes and disorders of conditions. We also know that there are certain fundamentally human traits that need to be studied in terms of cognitive capacities, things like language. So when we have brain injury and language disorders that affect these processes in the brain, we can study the brain to understand what it's doing as a function of dysfunction. So we learn a great deal about what the brain does from these studies of pathology, and taken together with all these direct lines of evidence from the animal studies and from human lesion studies. And there have been a number of patients that have been studied that have had disorders of social cognition caused by focal brain injuries, and things like damage to the frontal cortex. And several brain regions stand out as really important for these normal social interactions with others. And these can include a wide variety of cortical regions, such as the frontal cortex and the somatosensory cortex, and other areas that are associated with simulation and empathy. And by empathy, I mean sort of mirroring. And all of these and many other structures have been co-opted and sort of modified to subserve specific demands that are associated with increased social complexity. And this is known as sort of the social brain hypothesis. And this includes things like mediating social approach in a variable dominance hierarchy, or processing social stimuli and relational information in terms of remembering which individuals are related to whom, and things like metacognizing the thoughts and the behaviors and the intentions of others. So a quick rundown of what certain brain regions do. We have the frontal lobe, which is involved in working memory, emotional control, decision-making, and aspects of personality. And when this is damaged, you have certain defects that relate to these different processes. Things like planning behavior become very difficult. Personality changes are common in people who have frontal lobe lesions. In the parietal lobe, we see that sensory integration and spatial awareness and perception can be affected when these areas are damaged. In terms of the occipital lobe, visual information and the processing and integration and interpretation of all of these different visual stimuli is rooted in the function of the occipital cortex. And the temporal lobe has a variety of interesting functions, including things like hearing and language comprehension, because this is where the primary auditory cortices are. But there are also structures of the limbic system that participate in things like information retrieval and stored memory. So in human evolution, we've actually seen a wide variety of modifications to certain regions of the brain. And these include things like the reorganization of the prefrontal cortex. Certain areas have become disproportionately enlarged relative to other areas. The parietal lobe has seen the expansion of the inferior parietal, which seems to relate to the increase in tool use. So facilitating tool use requires integration of a higher degree of sophistication in the inferior parietal lobe. In primates, we've seen a great deal of expansion and elaboration of internal circuitry in the occipital cortex that has to do with adaptations for visual predation, color vision, and food selection, and things like this. And in the temporal lobe, we have regions such as the amygdala, which deals with fear and with emotional reactions to stimuli, which have been co-opted for things like facial recognition. And in the amygdala, we know that we have a greater number of neurons, and that the amygdala itself is enlarged relative to brain size, and there may be increased connectivity in this region, as well as the hippocampus, which deals with spatial memory and encoded verbal memory and all other aspects of memory. So what I do is primarily post-mortem neuroanatomy, and specifically neurohistology, which means that I look at the brain of individuals who are deceased, and I process them, and I stain them, and I look at things, and I count things, and I tell you what's different. So what we do is a process called cryosectioning, where we freeze chunks of the brain that we take out, and then we cut them into very, very thin slices using what looks like a really, really advanced deli slicer. And then we stain these with a variety of different proteins in order to look at the distribution of different cells and other elements, including neurotransmitters and proteins in actual tissue from the brain. And this can be done in postmortem tissue, and there are a lot of things that you can do in postmortem tissue that you can't examine in living individuals or otherwise. So at the cellular level, a lot of these histological examinations have yielded really valuable information regarding the evolution of the prefrontal cortex, for example, which is involved, again, in things like executive function. And what we know from looking at the gross anatomy is that the human frontal cortex is not disproportionately enlarged in size. However, the prefrontal cortex does show certain signs of reorganization, including the enlargement of certain areas and the reduction in size of other areas. One of these areas that seems to be enlarged that seems to be modified includes the frontal polar cortex, or Brodmann's area 10, which seems to exhibit a differential increase in the spacing between cells, known as neuropil spacing. So in between individual neurons, what we have is the branching of the neurons and all of the connections that they form between them. So if there's more space, there's more room for these different connections. So decreased neuron density means that there's significantly more room for connections to form between cells. And this greater branching of individual neurons seems to reflect some sort of difference in the way that communication occurs, both across the brain and in the local processing unit, known as the mini column. So I've talked a lot about the cortex, but the cerebral cortex isn't the only place where evolutionary modifications have occurred, but it does get the most attention due to the fact that it's uniquely derived in mammals. So, and this is also part of, the focus on the cortex seems to follow logically from sort of the history of evolutionary neuroscience as a discipline. And in the 1960s, an evolutionary neuroscientist by the name of Paul MacLean introduced a taxonomically informed model of the triune, or three-part brain. And the structures that are found in the ancestors of the most primitive or conserved vertebrates were assigned to this designation of the reptilian brain. And these are structures that are involved in really basic homeostatic mechanisms, as well as some basic survival and the orchestration of motor movements. And structures of the reptilian brain include the basal ganglia, which derive early in development from the floor of the forebrain, and the paleomammalian brain is seen in the middle. And this includes things that are regarded to be part of the limbic system, including the septum, the amygdala, the hypothalamus, the hippocampus, and the cingulate cortex. And altogether, the limbic system is responsible for motivation and emotion involved in feeding, reproductive behavior, and parental behavior. Lastly, this neomammalian complex consists of the cerebral cortex, which is found uniquely in mammals. And MacLean regarded this addition as the most recent instance of novelty in the evolution of the mammalian brain, which in turn is scaled up in human evolution, in primate evolution and in human evolution, which confers the ability for language, abstraction, planning, and perception. So in truth, all of these subcortical structures are very, very old, and they possess homologs, or things that are shared with our oldest vertebrate relatives. And it's fairly interesting to note that analogs of these structures seem to exist in non-vertebrates, and some may be as old as around 500 million years old. Limbic structures are also found in a wide variety of non-mammalian taxa, and the neocortex is, of course, the defining mammalian trait, but it was likely present in a small part in some of our mammalian ancestors. So also, some non-mammals, including things like birds, possess a distinctly enriched pallium, which is this layered structure that's like a cortex, but it doesn't have the same layers or the patterns that are associated with cortex. But we know that bird palliums observes some very sophisticated cognitive functions, including things like tool use and really, really complex communication. So it's not just all about the cortex. We examined the limbic system in human evolution along with one of my colleagues, Dr. Nicole Barger. And in our lab, we compared the relative size as volume of certain subcortical structures across primates on the one hand, so all primates, and hominoids, or us and the apes. And we found that certain structures seem to represent these marked departures from expected values, which indicates that the reorganization of brain systems may be related to emotional control and regulation. So specifically, we found that nuclei of the amygdala tend to show evidence for reorganization, which is important because the amygdala is important for things like facial recognition and conspecific recognition. The human striatum, which I study, is involved in action selection and motor and decision making, and it seems to actually be significantly smaller than would be expected in a human in primate brain size. So this seems to pose this fundamental question about the computational implications that reductions and enlargements may have. So our further research is going to address some of the cellular composition of these regions to sort of clarify trends in the functional significance of these reorganized elements of the brain. Now again, the cortex makes up that outermost layer of gray matter that makes up the superficial aspect of the brain. And the gray matter is where the cell bodies of the neurons are, and the white matter consists of the fiber tracts that tend to connect brain regions. So gray matter is where your cell bodies are, and white matter is where the connections between different brain regions move. So we found some evidence for connectivity and reorganization being modified in human evolution, and local connectivity may in fact be increased in some regions due to the fact that there may be increases in cell branching that are reflected in decreases or increases in space between neurons in certain areas. So local connections between things may be modified, but also long-range connections include these white matter fiber bands seen here, and include things like the arcuate fasciculus, which literally connects the areas for speech and language perception and speech and language production. So I want to shift gears here a little bit and take some time to consider intraspecific variation, which is, of course, to say, how does human cognitive potential actually vary, and what does this mean for human evolution? Now, all of the people on this page are considered geniuses for various regions, but they exhibit a vast array of talents and different intelligences. And importantly, many of these individuals seem to have talents and abilities that outpace others that may occur as a sort of specialization. So artistic ability versus math and spatial ability and quantitative ability and things like this. Many people tend to specialize in one area or another. But many times what we see, there's some suggestion that many times we see among geniuses various impairments to cognitive functions, so specialization at the detriment of other functions. So it's very important to think and stop and consider how we define neurodevelopmental disorders, and to ask the question, are deviations from what is considered typical function inherently pathological? So should they be pathologized and treated, or viewed as normal extremes of the spectrum of human intelligences? And there's this growing movement of individuals who want to identify traits in human potentials along the lines of neurodiversity. So individual potentials may represent different strengths and weaknesses and ways that just reflect the spectrum of human potential. So this has gained a lot of traction among the community of individuals affected by autism spectrum disorders. And I probably don't need to spend a lot of time describing autism spectrum disorders because of its relatively high prevalence, which means that most of you in this room probably know someone who has been affected by an autism spectrum disorder. Autism spectrum disorders have a very high prevalence among the general population, and this prevalence may in fact be increasing. It was as much as 1 in 68, according to a CDC report in 2016, and we know that it's 5 times more common in boys at a rate of 1 in 42 than it is in girls. And we know that autism spectrum disorders are characterized by a very complex and variable behavioral and cognitive phenotype, which at its core reflects several characteristics which qualify it as autism spectrum disorder, including reduced social engagement, delays in the acquisition of language and language ability, and generally impaired communicative function and social interaction. So it's important to understand that autism spectrum disorders encompass a wide and broad spectrum. And on one end of the spectrum, you can have individuals with cognitive abilities that may in fact be enhanced. So high functioning autism is something that gains a lot of attention because of many individuals tending to have better than average or conserved abilities in quantitative skills and in spatial perception to the detriment of language function. Of course, this can extend all the way to very severe learning disabilities, but along the spectrum of variation, we see that autism spectrum disorders are actually an umbrella diagnosis which encompasses a wide variety of different variants. One recent and controversial theory that has been supported by some individuals includes this extreme male brain theory of autism. And of course, this suggests that autism is just an extreme or exacerbated version of the male brain, with language and social skills being seen as sort of more feminine or female cognitive faculties, and spatial and quantitative intelligence being seen as more male. And I always enjoyed discussing this with anthropologists because the title of the book is The Essential Difference, and exactly what it does is kind of essentialize gender and put into very rigid categorizations that designate things as more masculine or feminine. Though we know that among individuals, these potentials tend to range in ability, there's only limited support for this idea put forth by this idea of the fetal androgen theory, that it suggests that exposure to male hormones, specifically testosterone, in utero may predispose the brain to become more male by this definition, and thus more autistic. And it's true. From some studies, we've learned that increased testosterone may contribute to greater quantitative and spatial ability. However, these things are not definitive. Among advocates of the neurodiversity movement, individuals like Temple Grandin, seen here, tend to assert that autistic-like behaviors may convey some sort of specialized ability. And Temple Grandin, of course, said, I think autistic brains tend to be the specialized brains. Autistic people tend to be less social. It takes a ton of processor space in the brain to have all the social circuits. I mean, after all, the first stone spear was not designed by the totally social people. So this idea that there may be trade-offs in cognitive strengths may gain some limited support. And individuals may have the ability to have their abilities seen as just a part of the normal spectrum. And if this is indeed a spectrum, what would be on the other side of it? So I'd like to introduce you all to a disorder known as Williams syndrome, which is a fascinating variant that has some very interesting features cognitively and behaviorally that can help to serve as sort of a counterexample to autism spectrum disorders. And I have a short video for you.
Think you can be born too friendly? I don't mean a baby who smiles a lot. I mean children who simply have no fear of strangers, who can't even imagine anyone ever wanting to do them harm. Meet some kids who I hope will fill your hearts the way they did ours. They're living with an extreme medical condition that is both a blessing and a curse.
Hey guys, this is the story I laugh.
Imagine a place nestled in the woods, where music fills the air, where you can dance to the beat of your own drum. And for some reason, everybody here wants to be your friend.
Nice to meet you, bud.
Nice to meet you. You're great. You're great. You got great hands.
Hi, Chris.
Hello, buddy. How are you?
I'm really watching on ABC News.
Oh, thank you. Thank you.
What's your favorite color and what's your favorite TV show?
Kids love to ask questions, but not as much as these kids.
My favorite color is blue. I have met Barney the dinosaur.
Where do you live?
I live in New York City.
Do you have any sons or daughters?
I do.
Wow.
I have 2 daughters and a son.
And kids love to make friends, but Not like these kids.
What nationality are you?
I'm Italian. Oh.
How do you, buddy?
They look a little different, but what makes them special is a rare condition called Williams syndrome. And here at a special camp in Michigan, these kids have gathered for a week of learning, fun, and togetherness. It may be the most welcoming environment in the world.
My favorite thing to do is come here and hang with my friends.
Who likes hugs?
Something about, oh, I love
I do.
when people give you a big, huge, giant hug, you feel really loved. It's very nice to do to people.
The face that they like to put forward is that one of joy. As soon as you smile at them, you are their friend. You're no longer a stranger.
Terry Moncava started this camp back in 1994. Her 24-year-old son, Ben, a camp counselor, was more than happy to give us a tour.
There's a lot of things you can do. There's like fishing, there's boating.
Hello.
Williams syndrome is a genetic condition affecting 1 out of every 7,500 newborns. About 25 genes out of 25,000 are deleted in the middle of the 7th chromosome. That little genetic deletion creates a whole constellation of symptoms, including exaggerated, vaguely elfin features. The children also have hypersensitive hearing. That means, on the one hand, noises like a parrot squawk can terrify them. On the other, they show a remarkable affinity and, in some cases, talent for music. Ben is an accomplished drummer. And whenever you're with Williams kids, put on the tunes because they are ready to get down. But by far, the most fascinating aspect of Williams is a complete lack of social inhibition.
I have a question to ask you.
All right, let's hear it. What is it?
Do you like girls?
I do like girls.
It's like a friendship gene, a happy gene.
Yes, a little bit like that. We're trying to understand what are the underlying cognitive and brain mechanisms that explain the social interest, the social exuberance that we see. Do you worry sometimes?
Dr. Helen Tager-Flussberg of Boston University has been studying Williams for almost 20 years. Like other researchers, she runs many of her experiments during the biennial conventions of the Williams Syndrome Association, which, as you can imagine, are rather spirited affairs. You might think all kids are this warm and open, but look at an experiment that proves otherwise. The tester pretends to hurt her knee. A normal 2-year-old does nothing and actually keeps her distance. The Williams kid does the complete opposite. Moves in, tries to comfort, even rubs his own belly in empathy. Just what I wish my kids would do. That hunger to help was evident when we met 8-year-old Ashlyn Bear.
I want to help everybody when they get sick or get hurt or if someone's feelings are hurt.
So you're just you want to be a helper. Anybody, anything.
Yes. It feels like an honor to me.
We aspire to have a little bit of Williams syndrome in all of us. And I think when you meet a person with Williams syndrome, it makes you think about how you respond to other people.
Look how 11-year-old Eva Stoll is drawn to every dog and every owner she sees.
May I pet your dog?
The inhibitions that I have naturally, they don't have.
Right.
But is that a blessing or a curse?
I don't know. I think it's more of a blessing. I really do.
I think it's the rest of us who have a disability because we don't know how to be as kind and as open as they do.
Well, wouldn't it be great if the whole world felt that way?
So as you can see from the video, what you have in many ways is sort of an antithetical phenotype from what you see in things like autism spectrum disorders. So what you have is individuals who are very likely to engage in social approach. And beyond that, you have some other interesting phenotypic features. So again, Williams syndrome is a really rare genetic disorder that's characterized by a hemizygous deletion of about 26 to 28 genes on the 7th chromosome. And we'll unpack that a little bit. But in general, these individuals tend to have a tendency to view social stimuli as inherently more rewarding And a typical statement from somebody with Williams syndrome would be that everybody in the world is my friend, and a stranger is just a friend that you haven't met yet. There are no strangers if you have Williams syndrome. So in terms of phenotypes, we see a wide variety of effects caused by the genetic deletion on a bunch of different brain and body systems. So the disorder itself involves as many as 26 to 28 genes, and patients are expected to have multiple symptoms due to this deletion, and it's a haploinsufficiency effect, which means that the other allele of the gene that's left in the genome doesn't produce enough of a target protein to function normally. So individuals with Williams syndrome tend to have general global reductions in total brain size, in addition to certain deficits of visual spatial function. And in addition to sort of reduced global IQ, there is this hypersociability and also enhanced or preserved linguistic capacity. So interestingly, it's been studied across cultures, and what was found was that the use of descriptive vocabulary and the tendency for social approach seem to vary in culturally predictable ways. So this was a study that compared Japanese children with Williams syndrome to American children with Williams syndrome. And in general, this perception of being slightly more reserved is consistent across the Japanese sample. But the incentive saliency of the approach, or the tendency of individuals to engage in social approach, seems to vary in these culturally predictable ways, which is very interesting. So in terms of the etiology of Williams syndrome, what happens here is a failure when the crossover occurs. So when crossing over is what happens when gametes are produced. And each chromosome the chromosomes are duplicated and made copies. And so everything is rearranged, which contributes to the natural variation. And when a piece is deleted, you have a small deletion. And the deletion in this case is around 26 to 28 genes. And it's interesting to note that these specific genetic deletions seem to correspond to this really interesting characteristic behavioral phenotype. And the deletion itself implicates quite a few genes that are associated with development of the brain. And particularly interesting is that the deleted region has been compared in typically developing humans and extant African apes and monkeys that show evidence for a high rate of de novo mutations and sort of a dynamic evolution at this locus. So this is, in other words, a hot spot for recent human evolution and sort of change over time. So there are a variety of genes that are implicated in the deletion, and the first and most important is the principal gene that we use to genetically diagnose Williams syndrome, and this is the gene for elastin, which is a principal structural protein that occurs in tissues of the body, which contribute to things like the heart defect that is characteristic of individuals with Williams syndrome, which can be fatal if not treated. It's called supravalvular aortic stenosis, for those who are interested. But there are also brain-specific genes that are deleted in Williams syndrome that seem to have some very important functions, including this limb kinase 1 gene, which corresponds roughly to spatial ability and motor integration, as well as CLIP2, which is something that deals with cytoskeletal elements of the neurons, so the actual proteins and structures that form the backbone of neurons is impaired. There's also GTF2-IRD1, which is a gene that is involved in the craniofacial and skin variations that are caused. And GTF2I, which is a gene that seems to participate in immune function and may have something to do with the increase in glial cells that we see in the brains of individuals with Williams syndrome. There's also things like syntaxin, which is a gene that encodes a protein that allows individual neurons to communicate with each other using neurotransmitters. And this may be the source of the reductions in IQ in Williams syndrome. And there are several other not brain-specific genes that are important for a variety of reasons, including this BAZ1B gene, which has to do with the body's regulation of vitamin D, which is a systemic effect that may have some downstream effects in terms of neurological function. And NCF1, which has to do with the regulation of blood pressure and blood volume. So if that wasn't enough to get you really interested in Williams syndrome, there's something else, another disorder called DUP7, or duplication of the 7th region, the autism, or the Williams syndrome region on the 7th chromosome. So this very small subset of the population of autistic patients has been found to have this duplication in the Williams syndrome region. So remember, if you will, this process of crossing over happens in a way that leads to the duplication of a small amount of genetic material. And this tends to coincide with this autistic behavioral phenotype, including language delays and reduced social engagement. So what we're looking for overall in terms of understanding neurological disorders and neurodevelopmental disorders and picking them apart to figure out what processes are impaired, we're looking really for something called psychiatric endophenotypes, which are these sort of broad categorizations of closely linked traits that may correspond to impaired function in certain parts of the brain. And this can include things like impulse control, social inhibition, and social approach and behavioral response inhibition, which is impaired in Williams syndrome, and may implicate regions of the basal ganglia, for example, and the communication between the frontal cortex and downstream targets in the limbic system. And additionally, things like language learning may represent this other sort of module of cognitive function that can be studied and understood as sort of a psychiatric endophenotype. And furthermore, social engagement and empathy and attachment and these sort of pro-social facets of cognitive engagement are also very important. So what we're trying to do here is sort of reverse engineer cognitive potentials to figure out what strengths and weaknesses individual disorders may represent that may have some sort of biological underpinning that we can better characterize and better understand. So it's important, though, to understand constraints. And many of you have probably encountered this quote before. And I'd like to disabuse you of the notion that it was actually Albert Einstein who said it, because it wasn't. But the quote says that everybody is a genius, but if you judge a fish on its ability to climb a tree, it'll live its whole life believing that it's stupid. And again, this is probably not something that Albert Einstein said, because it's actually kind of interesting to note that Albert Einstein was a slow learner who was very late to speak and read and had some linguistic delays that may sort of represent this trade-off between linguistic and other abilities, including math and spatial skills. But I'd also like to throw in there that some fish do climb trees, and this is a mud skipper. So take it for what it's worth. So finally, what we want to consider here is whether or not human cognitive potentials are fixed. And though we're born with a vast majority of all of the neurons that we'll ever have in our entire life, the connectivity between them is constantly being remodeled and modified by specific input to each cell. And it does so in such a way that areas that are more consistently stimulated, cells that are more consistently stimulated, are more likely to receive connections and stronger connections. And then there's this thing called mini-column spacing that involves the space between neurons. And this continues to grow in humans into adulthood, which suggests that the elaboration of local connections may be driving some of the differences in morphology here that relate to the way that input-dependent learning is occurring and the modification of the brain is following from that. And this continues all the way into the third decade of life. And it continues to grow in humans. And this effect can be magnified in the human prefrontal cortex, as you can see here. And this is the really important site for learning and decision making. So thinking more about plasticity, we should consider the fact that myelination also seems to be an important feature of how human cognitive plasticity, or brain plasticity, is enhanced. And we know that humans exhibit slower myelination across development, which seems to continue all the way into early adulthood and well into the third decade of life. So your brain is still not fully baked. You're still requiring the inputs, and all of these inputs are going to continue to modify them. So in terms of specialization, we know that from performative studies, training of expertise for skills causes predictable and interesting changes in the brain, including things as complex as the growth of the hand area of the cerebral cortex in musicians. So what you do with your brain really does matter in terms of how it's going to look and how it's going to function. So maybe it's best that we start considering the idea of reconceptualizing the spectrum and breaking down into these categories where we fall in terms of different cognitive strengths and weaknesses. And through that, we can approach this sort of neurodiversity and understand that we all sort of fall along different lines in terms of our cognitive abilities, and that these just represent sort of a natural variation in terms of humans. Now, it's also important to note that a variety of research and education has indicated that attitude is very important. So those who tend to believe that skills and talents are naturally born in, and you're either born with it, and you can either learn music or you can't, tend to have a harder time with skill acquisition than do those individuals with a growth mindset, which would encourage the effort expended and to sort of increase the skills and talents by successive efforts and putting effort forth. So in the education literature, this idea of fixed versus growth mindsets is increasingly important. And it seems to correspond very well to the fact that what you do with your brain matters for the way that it develops. So in the future, our interventions for cognitive and social deficits, as we're portraying them, should include things like educational and behavioral interventions that should develop existing strengths and improve upon sort of key adaptive skills that utilize this growth mindset. Additionally, these efforts tend to be more successful when they're skill-focused, so have a specific task in mind. And we can start to approach things like brain training to utilize our natural plasticity to increase our own abilities. But a focus on early intervention and novel approaches is absolutely essential, because there can tend to be these sort of critical periods for developing different skills. Language is chiefly among these that has sort of a sensitive period when you really need to be working towards expanding vocabulary and understanding phonemes, things like that. So in conclusion, brain evolution itself is very nuanced, and it's best studied from multiple levels of investigation. So indirect versus direct evidence of the evolution of the brain is important to integrate these elements. And finally, neurodevelopmental disorders provide a fascinating opportunity to observe sort of natural experiments in the broad spectrum of human phenotypic variation that can inform us better in terms of how these things affect brain growth and development in both neurotypical individuals and those who fall at every end of the spectrum. With that, I'd like to acknowledge my co-authors and my fabulous colleagues.